Good afternoon. Today we'll be working on ATP and chemiosmosis, which is part of cellular respiration. Before I forget though, I wanted to make sure, that I pointed out something as far as it relates to practice free response. If you go into our Canvas page, off to the left, there's a spot that says Files. And in Files, there's a file named AP Bio FR Free Response, referenced in YouTube vids. So I'm referencing these in YouTube vids now. When you open it up, one of these yellow boxes should be open, but if not, you can just click on it. And like here, I can minimize it. And then if I click on it, it'll pop back open. It sort of explains how the yellow boxes work. And the way that they work though is here's a yellow box. So I'll click on it. This says 2010 number 2, so 2010 not number 1, but question number 2. That one's number 1. I'm not sure how well you can see that, but this is number 2. It says 10 points, 22 minutes. So back in the day, this was a 10-point question, and you got technically 22 and a half minutes to do it on average. The topic is enzymes, and when should you do it? You should complete it after review video number 3. So you watched review video number 3, now try this free response. I have all of the anticipated free response that we're going to look at for the rest of the year. Now that might change on April 3rd after College Board releases stuff, who knows. But basically I'll just scroll to the next one in line. I've got them sequentially based off of our reviews. So for example today, this one says, this is 2009, number 2, A, B, and C only, 8 points for ABC, about 18 minutes. Topic is ATP and chemiosmosis. And you should do this after review video number 4. Make sure you don't stop there though. Look ahead to the next one, because this one also says that you would watch it, or you would complete this one after review video number 4. Do I have a third one? I click on this and it's like, oh no, that's after review video 5. Okay, so I've got 2 today that I would work on. When you're working on these, ideally you would sit down, time yourself, writing complete sentences, give yourself 22 minutes or 18, however many minutes it suggests. In reality, that might not be doable. I don't know how busy you are or how serious you are about the real AP Bio test. And also this year we have that oddity of COVID, so we're doing a 45-minute test. So our strategy might change a little bit after April 3rd when College Board is supposed to tell us more. But if you can't do that, I would still time myself, get out a blank sheet of paper and put down bullet points. Kind of like when we'd have rubrics on our free response, like, okay, ATP synthase is hollow and it spins. When spins, joins, when spins, ADP plus phosphate equals ATP, just like a few hit points. Don't just sit there and mentally be like, I've got it. Jot down the important things that you think would get you points. If there's a calculation, actually try the calculation. You can use calculators on the test, assuming that doesn't change. Okay, I think that closes us out on that. By the way, on the future ones, I don't know exactly, like when we're in this particular unit right here on cell communication, that's a few weeks from now. So I don't know if that's after video 10 or 11 or 12. So it says when, TBA. So every week and a half, 2 weeks, I'm gonna update this. In Canvas, it'll still be AP Bio FR referenced in YouTube vids, and I'll update it so that you can kind of see the next week or so. So I'll start filling in all these to be announced. I will actually announce them. All right, so what we want to look at is the chemical structure of ATP. When I look at ATP, I have an adenine. What is that right there? It's a pentagon with an oxygen in the corner. So it's got to be some type of carbohydrate. On this one, this is a little bit of just fun facts. This is ribose. I know that because right there, I have an OH and an OH. If I got rid of that O, then now it's deoxyribose because I just deoxied it. So there's an adenine, there's a ribose, and there's a phosphate. So isn't that a RNA nucleotide? Isn't RNA A, U, G, or C? There's adenine, a ribose sugar, and a phosphate. Yeah, that's an RNA nucleotide. But if we slap a couple more phosphates on it, it's adenosine. Adenine and ribose together are called adenosine. And then I have 1, 2, 3 phosphates. So you might remember from your chemistry class that phosphate, gotta adjust now. The phosphate looks like that orange drawing I have there. So if I'm looking at this phosphate, hang on, I started at about 11:35. That's how I time myself. So I've got a phosphate molecule, a phosphorus in the middle, 4 oxygens around, so you learned that it was PO4. And then your teacher also told you, if you had to memorize like your polyatomic ions, that it had 1, 2, 3 negative charges. And when phosphate is on its own, that's true. It has 3 negative charges. Over here, if you were to Google, or if you're just looking in either your enzyme notes or your cellular respiration notes, we have this picture of ATP. If you were looking at it, you'll notice that that phosphate doesn't have 3 negatives. It's only got 1. Well, why is that? That oxygen up there doesn't have a negative. Why not? Well, oxygens, to be stable, are supposed to have 2 covalent bonds. So that oxygen is totally stable in order to be, I should say, neutral instead of stable. So to be neutral, then it has 2 bonds. That oxygen does not. But if I start joining it to another phosphate, like this, now that oxygen has 2 bonds, so that negative goes away. And this guy's got a negative. This guy right here would have a negative. But if I chemically join it to another phosphate, like this, I realize we're getting rid of, there's not like an oxygen and an oxygen, so they're like that oxygen is shared between that phosphate and that phosphate. So I continue like this until I'm attached to adenosine. And anytime I have an oxygen with only 1 covalent bond, it'll have a negative on it. So that's why typically your teacher told you that phosphate has 3 negatives. This phosphate right here has 2 negatives, but these phosphates that are kind of in the middle only have 1 negative. That's why. Okay, why do we care whether or not they have negatives? This is the essence of ATP. ATP is the energy molecule for all of life. If I go through and I look at that chart of life, as I call it, I can zoom in and I've got all these chemical reactions going all over the place. I know that we've seen this chart many times in class. I've even used it in 1 or 2 of the videos so far. I think the last video, talking about aspirin. So in this chart, there are chemical reactions just going everywhere. But then when I finally get down to the very bottom of the chart, there is one final output, ATP. So all of these reactions can feed into making ATP. It's not just glucose. We can also take fats and shove them into the Krebs cycle and start doing the Krebs cycle and electron transport chain and making ATP also. So we always focus on glycolysis, Krebs, and electron transport. But we also convert fats into ATP as well, not just glucose sugar. So why is ATP the special molecule? In plants, fungus, humans, it's not the only one. We can also use GTP. We can also, plants use sunlight hitting chlorophyll to provide energy to power chemical reactions. So it's not the only thing, but it's still the primary thing. It's almost like electricity. Everything in your house is pretty much running off of electricity. Maybe your car is not, or something like that. Maybe it's running off of gasoline. But even then, most of your car is still running on. You're converting that into, anyway, I digress. So why ATP? Well, what makes ATP special is its chemical structure. So what is odd about ATP? Okay, the shape is kind of weird. Phosphate with negatives, phosphate with negatives, phosphate with negatives. So I have an adenosine with 3 negatively charged phosphates joined. Does that seem stable? Well, no, a negative thing bonded to a negative thing bonded to a negative thing, they should be repelling. And so at first you might say, well, it's stupid to make ATP the energy molecule for all of life if it's highly unstable. But then you think of like gasoline, really good energy source, highly unstable. If I had a cup of gasoline, I don't know why, and I shot a spark at it, a spark might be enough to ignite that. Paper's flammable. If I shoot a single spark at this piece of paper, I doubt it's going to burst into flames. So gasoline is highly unstable. kerosene, you think of lighter fluid, you think of propane. So most of our energy sources are highly unstable, right? Same with ATP. So the fact is, the bonds between these phosphates are highly stressed. So all these bonds in here, if I've got a phosphate that's negative, a phosphate that's negative, and a phosphate that's negative, 1, 2, 3, like this, then the bonds between them are stressed because this negative phosphate and this negative phosphate are repelling. I always give the example of if this elbow's a phosphate and this elbow's a phosphate, then if this is the bond between them, then this negative and this negative, they're repelling. They're trying to pull apart. Now they're currently unsuccessful because the bond, the glue, the covalent bond holding them together, the shared pair of electrons holding them together is stronger than the repulsion. Just like if I had two magnets and if I don't touch them, they repel. But if I want, I'm strong enough, I can like hold them together. They're still trying to repel each other, but their repulsion force is smaller than my force pushing them together, so they have no choice. And I know they don't have a choice anyway, they're chemicals, so they don't have brains, but you get the idea. So they are repelling each other. That's actually the genius of ATP. So how much energy would it take to break this bond? Again, phosphate number 3, phosphate number 2, phosphate number 1 is over there, but I ran out of elbows. So they're going like this. How much energy is it really going to take to break this bond? A lot or a little? A little. So it takes very little energy, and once I break it, what happens? They fly apart. So if I were to look at a free energy diagram or like an exothermic graph of ATP, here you go. I'm going to use this for something else in a second. Maybe a typical chemical reaction looks like this. A typical exothermic reaction. And I am going to rehash a joke that I used in class earlier this year. But with ATP, now, how will the graph look different? Well, does it take a whole lot of energy to break this phosphate from this one if they're already repelling? No, so it has a low activation energy. Just like gasoline, a small spark is all the activation energy you need. So instead of having a giant bump, it's got a teeny tiny bump of activation energy. And when it does, it releases energy. How much? A ton. Off the chart. So that's why ATP is special. So in summary, ATP is adenosine triphosphate. If you had to classify it as one of the 4 biomolecules, lipids, carbs, proteins, nucleic acids, you'd call it a nucleic acid. It's a modified nucleic acid. Add a couple of phosphates onto adenosine monophosphate, an RNA nucleotide. And then what's special about it is that it's unstable. What's causing it to be unstable? All the phosphates are negative. Negatives repel each other. Well, that's the genius because now it takes very little input energy to break the bond. So I don't spend a ton of energy trying to get energy out of ATP. And when I do, I release a ton of energy. So that's why ATP is great. Just like gasoline. 1 gallon can shove my SUV down the street. My SUV weighs, I don't even know how many thousands of pounds. But it can shove my SUV down the street 25 miles, something like that, from a gallon of gas. So it releases a ton of energy, and it takes almost no energy to get it burning. So that's why ATP is special. Another thing that we want to look at before we get too deep into this is hydrogen, oddly enough. Hydrogen has a 1 up here for an atomic number and a 1 down here for a mass number. Atomic mass would keep the decimals, that's like an average of all the isotopes, but mass number is we just keep it a whole number. So 1. What does that mean? That 1 means I have 1 proton, and when I'm looking at hydrogen on a periodic table, it's neutral. So if I have 1 proton, I better have 1 electron so that I'm neutral. So I have 1 proton and 1 electron. So let me write that in up there. 1 proton, 1 electron. My mass number is my addition of total protons plus total neutrons. Well, if it's 1, that means I have 1 proton and I must have 0 neutrons. So hydrogen doesn't have any neutrons. Quick little chemistry trivia. The reason for that is it doesn't need them. Everybody else on the periodic table needs neutrons. That's because the function of a neutron is to act as a spacer. You might have heard that neutrons stabilize the nucleus. That's true, but like how? The way it works is if I've got helium, for example, in the atom of helium in the nucleus, I've got a proton and a proton. Helium has a 2 up there. So I've got a proton and a proton. What happens if I put protons close to each other. They're both positive, so what will they do? They'll repel. Well, are they close together when they're in the nucleus of an atom? Yeah, that's kind of the closest thing that we know of, is like all within an atom. Where? Within a nucleus. That's a tight fit. So what do? You put neutrons in there to act as like spacers. So they space them out just enough so that the nucleus doesn't sort of repel itself apart. And hydrogen has one proton, so it doesn't need a neutron spacer. Everybody else on the periodic table, if I put a periodic table up here, everybody else would have neutrons because they need them as spacers between the protons in the nucleus. Okay, so hydrogen. Regular old hydrogen is a proton and an electron. A hydrogen ion, H+, when something becomes an ion, that means it gained or lost electrons. It can't gain or lose protons. If it gains or loses protons, then it's no longer hydrogen. Like helium is helium because it has 2 protons always. Carbon is carbon because it has 6 protons always. Now it might be able to gain or lose. It's usually not ionic. I guess it's carbide or something if it is. But if it were to gain or lose electrons, it might become positive or negative or whatever. But it can't gain protons, because if it gains a proton, it's now, what is that, nitrogen, I think? If you suddenly have a 7th proton. So protons, number of protons decide who you are. Are you oxygen, nitrogen, whatever? The number of electrons will determine whether you're neutral, positive, or negative. Are you an ion? So hydrogen ions become hydrogen ions by gaining or losing electrons. Well, I've got an electron. If I became H+, that means I lost that electron. So a hydrogen ion is just a proton. That'll be important in a moment. Right now you're like, what am I learning here? But that'll be important in a moment. A hydrogen ion is the same thing as a proton. Okay, so with that in mind, let me check my time really quick. Yes. Okay, so with that in mind, let's look at chemiosmosis. So mitochondria, you might see a picture like that typically. Here's some microscope images. Mitochondria, there's mitochondria, there's a mitochondria. You'll notice that in mitochondria, here we are focused on just a single mitochondria. Most mitochondria are kind of cylindrical, and they aren't crystallized. They kind of move and sort of blob around. I know that's not a real verb, but should be. So we always see them as like cylinders because we cut this way. But if you cut a cylinder this way, it's kind of more like a circle. So that's what you're seeing here, is you're seeing one cut down the middle, but they usually are kind of elongated, kind of like gummy worms, except they're needed for life. Here's another picture of mitochondria, a few of them, the red guys. And what you'll notice about mitochondria always is that they kind of always have those little teeth-like structures, tooth-like structures inside of them. So this is my mitochondria. It has the tooth-like structures. And what was this membrane made out of? That's right, phospholipid bilayer. Both of these membranes are made out of phospholipid bilayer. So mitochondria have a double phospholipid bilayer. First you're like, wait, a phospholipid bilayer, it's a bilayer, it's already double. Yeah, that is a phospholipid bilayer. That's another phospholipid bilayer. So I have 1, 2 phospholipid bilayers. That guy is a phospholipid bilayer, that guy's a phospholipid bilayer. Okay. One thing you might remember about phospholipid bilayers, do things that just slide through. Sometimes I have substances that can just slide through diffuse from outside to inside or from inside to outside, high to low concentration. Things can diffuse through phospholipid bilayers. What types of things generally have an easier time getting through? Polar or non-polar? Small or big? Well, obviously small. Small things can squeeze through between these phospholipids easier than large things. Remember, that is fluid. One of those tails of the phosphate is typically saturated and one is unsaturated. So the phospholipid bilayer is fluid. They're kind of shuffling around. If this is the outer leaflet of phospholipid bilayer, the caps are the phosphates, and these are the tails. They're like sliding around laterally. The inside part, where it's facing down, is also sliding around laterally. So your phospholipid bilayer looks, I don't have quite enough pens, but something like that. So things can slide through if they're small enough and if they're non-polar enough. Larger things have a tougher time getting through. The more polar something is, the more likely it doesn't get through. That's also important. So why was this inner membrane folded? More surface area. So just like the little demo I did in class, let's say I give you this piece of paper and I say I want as many purple dots as possible on that piece of paper. Actually, let's do this. I give you this piece of paper. I say I need this post-it note to fit in my pocket. Okay, cool. And on there, put as many purple dots as you can. These purple dots we're going to find represent ATP synthase and proton pumps. Okay, so I don't know how many dots I can fit on here. Let's say a couple of hundred. I fit it in my pocket. Now let's say I have a couple of hundred ATP synthases that can make ATP. Or I could say, okay, this piece of paper. I can fit more dots on this bigger piece of paper. But now how do I get this if I put more dots on there? How do I get this piece of paper into my pocket? I just fold it carefully. There you go. So that's what we're doing whenever we say in class that if I fold this inner membrane, it's increasing surface area. I can now have a 8.5 by 11 sheet of paper, and if I wad it up, I can fit it into my pocket. If I were to stack pages, we'll see this in photosynthesis. If I were to stack pages, if each page is 1 square foot, pretend, and this thing is 1,400 pages, then that means 1 square foot of surface area, 2, 3, 4, 5, 6, 7, 8, 9. I have 1,400 square feet in here, right? A 1-bedroom apartment might be 600 square feet. So more square footage than an apartment, but it fits in a small space, a book. Okay, so now I've got this inner membrane. It's folded so I can get more of this stuff on there. That's why I want more surface area. Why does the outer surface area not matter as much? I don't give much of a crap about the stuff that's on there. I care about this stuff on that inner membrane. We call that inner membrane either the inner membrane or cristae. The name is not that important on the AP Bio test. The function of why is it folded matters more. Okay, this space in between here we call the intermembrane space. That's kind of long, so I'll just put intermembrane space. Intermembrane. Inter means between. So there's a membrane, there's a membrane. This is the space between the membranes. So it's an intermembrane space. And then out here, this area is called the matrix. Okay, so this guy right here is ATP synthase. ATP synthase. Let's think of the name for a second. ATP synthase. It ends in ace, so it's, yes, an enzyme, and its job is to synthesize ATP. ATPase breaks down ATP, ATP synthase builds it up. Remember in the previous video, anabolic, catabolic? ATP synthase is anabolic, ATPase is catabolic. It breaks down ATP to release energy. So we're trying to build up energy. Well, how does it do that? Basically what happens is anytime we want to release energy from ATP, we sort of like I was showing you before with this analogy, we're going to break off a phosphate. So typically we just break off one phosphate and we're left with adenosine di. Di means 2. I now have 2 phosphates. We're left with ADP. So if I break that off, I now have adenosine diphosphate plus a phosphate out here. Okay, whenever I do that, I release energy, and that's how I move around and everything else. My body converts this energy of the phosphate flying off into movement or thought or whatever. Powers active transport, stuff like that. Okay, so now what I do is I basically, if I use an ATP, pretend that this is an ATP, I break off a phosphate, so now this is ADP, adenosine diphosphate. Now it's ATP, adenosine triphosphate, tri, like that. That's what we do. We just keep recycling the same ADPs and phosphates over and over again. It turns out as long as I eat food and get calories from it, then I get the energy to take a phosphate and shove it back on there. It takes energy to do that because this phosphate is negative. And if this is ADP, that means that there are phosphates on the end that are negative. So it's like, hard to shove that on there. Negatives and negatives. ATP synthase is able to do that. How does that happen, you say? Well, ATP synthase is a molecular motor. So let's say you have a tube, like these paper towels right here. It's hollow. ATP synthase is hollow. ATP synthase also spins. So ATP synthase is hollow and it spins. Now, I'm showing you a slightly less realistic, slightly less complicated version of how ATP synthase works. But I'll have this is from DNA Tube running. If you just Googled, even on YouTube, if you just Googled ATP synthase, you'll eventually find this video. So this is showing kind of the more full strength version. I'm going to simplify it some. This is my hollow tube, and down here I have these other proteins. This is kind of what I'm drawing, the hollow tube and then these little proteins. These little proteins rotate, so relative to the tube. So you can see in the video how the tube part is kind of rotating, and as it rotates, what's happening is I take an ADP and a phosphate and slam them together. So I'm going to pause this for a moment. You can watch it on your own if you like. I just don't want you to be distracted, even though I know you can rewind. So essentially what's happening is if I can get this thing to spin, as it spins, it will, ATP synthase that is, it'll shove together an ADP and a phosphate. So think of it like this. What happens is it gets kind of twisted. You feed ATP synthase an ADP. So it grabs an ADP and a phosphate, and it sort of like twists, and then like slams them together. It builds up tension by twisting. This little part right here running down the middle. Here's the hollow part. You can see things flowing through, and as things flow through, those things are protons. As things flow through there, that will force this to spin. As it spins, it's building up tension. So this ATP synthase builds up tension, kind of like if you have a Cheerio car and you put it on the ground and pull it back in, it's like zzz, and then you let go and it takes off, kind of like that. So it's building up tension right here in this middle part, and then these little green parts at the bottom will grab an ADP and a phosphate, you'll see them move, and they slam them together. So as something flows through me, protons, because I'm hollow, that'll make me spin, and as I spin, that gives me the energy to kind of torque like this, ADP, phosphate, pow, I just made ATP. Give me another one, ADP and phosphate, pow. Okay, so it's hollow. Why is it hollow? It spins. Why does it spin? It spins to join ADP and phosphate. And how do I get it to spin? Well, it's hollow. So whenever things flow through, protons flow through this hollow ATP synthase, like that, that'll force it to spin, and it makes ATP. Great. Okay, so the way that we want those protons to flow is this way. I want them to flow from the intramembrane space out into the matrix. Because if they flow the opposite way, then I spin the opposite direction. And instead of building ATP, I break it apart. I always give the analogy, I picture at the Expo Pen factory, somewhere there's a machine that takes the newly made Expo Pen and the pen cap, and maybe in theory, it spins and shoves them together. So as long as I'm going this way, I'm building Expo Pens. Make me run in reverse, and I pull them apart. So I want protons flowing this way, from the intermembrane space out into the matrix, from the, what I'll call the rectangle side to the 1, 2, 3 circle side. All right, so how do I do that passively? Well, which way will these protons flow? Will they flow out into the matrix, or will they flow into the intermembrane space? Well, it's passive transport. This is a hollow channel. So it's hollow, or acts as a channel, ATP synthase. So we basically just do diffusion. We flow from high to low concentration. There's a little bit more there. We also have repulsion. Since these are positive, they can kind of repel each other out if it's crowded with pluses in there. It's not just a chemical gradient, it's an electrochemical gradient. The chemical gradient is high to low. The electro part is charged particles kind of repelling each other. So it's an electrochemical gradient, or you might hear a proton motive force is built up over here. And so these protons will flow from high to low. And when they do, as they flow through there, this guy will spin, and as the protons flow through the hollow channel, ATP synthase will spin, and when it spins, it joins an ADP and a phosphate. Great. Well, that works for a while. These guys are diffusing. They were over here, now they're over here. Okay, now I've got 6 and 6. So what does that mean? I just reached equilibrium. Is there any net movement going this way anymore? No, there's movement, but for every proton that flows that way and makes an ATP, there's a proton that flows that way and breaks it apart. So I'm not getting anything done anymore. I'm at equilibrium, so no net movement. There's still movement, but no net movement. Okay, so that thing's not spinning. That means no ATP. I'm gonna die, but I don't wanna die. So now what I do is I engage these little boxes. These little boxes are called proton pumps, and they do exactly what it sounds like. They pump protons. When we go back and we review cell membranes, we'll look at like pumps. If something's a pump, it's a type of active transport. So a proton pump pumps protons, but it's going to require energy. Normally we get energy from ATP, but here we're trying to make ATP. So unless we can find a way to like burn a few ATP to make many, then we'd prefer to not use ATP to power our proton pumps. We'd prefer to not have an ATP powered, ATP making machine if we can help it. So these proton pumps are going to take those protons and shove them back into the inner membrane space, like this. So now I've got a bunch of protons inside. They'll keep flowing through ATP synthase. It'll keep spinning. It'll keep joining ADP and phosphate to make ATP. Great. But where do these guys get their energy? That's kind of interesting. From electrons. So I'll start way back here. I steal electrons. I do pyruvate. The Krebs cycle will eventually get into all of this. And I'm stealing electrons from my food. Electrons will power the proton pumps. So sometimes students get confused on that. What does a proton pump do? It pumps protons. What gives it the energy to do that? Electrons. Kind of like my lawnmower. What does it do? It mows the lawn. What gives it energy? Gasoline. My car, it drives. What gives it energy? Gasoline. So we're naming the proton pump off of what it does, not its energy source. Just like my weed eater, again. I don't call it the gasoline using machine, and my lawnmower the gasoline using machine, and my car the gasoline using machine. I name it based off of what it does, but then they're powered by something different. So these proton pumps, these 3 proton pumps, are powered by electrons. So I'll have some electron carriers that you might remember. NADH, FADH2. So they are technically called coenzymes, but their job is they are electron carriers. And so NADH, technically it's whenever we are looking at these, it's called NAD+, and then once it's loaded up with all of its electrons, it's called NADH. I'll explain that a little bit more later. You're like, where did the H come from? But it basically comes in as NAD+, grabs electrons and becomes NADH. This comes in as FAD and becomes FADH2. In AP Bio, that likely doesn't matter. In a college biology class, it could matter. So knowing the difference between those is probably like tier 2 or tier 3 information in AP Bio. If you just see NADs and FADs and realize they're electron carriers, you're probably okay. I think of it as that one looks more full. To me, NAD plus versus NADH, that one looks more full. So it's full of electrons. FAD, FADH2, that's bigger looking to me. It's got more letters. So to me, it's full of electrons. So FAD is empty-handed. It grabs electrons, now it becomes FADH2. NAD plus is empty-handed. It grabs electrons and becomes NADH. Okay, so now I steal electrons from the Krebs cycle. There's also some NAD coming in from up here. NADH, for example. Bring that in also. And these NADH molecules, they go to that very first proton pump. And what they do is they drop off their electrons. Well, electrons can carry energy. At the simplest level, We mentioned this in a previous video. Think of it this way. When I shine light on a dark shirt, we said that electrons get excited and they go up in energy levels. And when they fall back down, they release energy. Well, you can kind of think of it like this. NADH is more complicated than that. It's not just a single atom. But still, overall, the overall energy levels do work like this. So NADH is carrying electrons at higher energy levels. It will then send them to this first proton pump. That proton pump will take those electrons and drop them in energy levels. Now again, it's more complicated. We aren't just looking at one atom, we're looking at molecules. So I'm simplifying it a little bit. You can get into like some pretty deep physics and chemistry, going into this, even some quantum mechanics a little bit. But, and that's beyond my pay grade for sure. But basically what happens is these guys are receiving them at a higher energy state. And then we hand them off to this little carrier protein, and then to another pump, and to a carrier protein, and to another pump. And along the way, we just keep dropping, we keep sucking out a little bit of the energy out of these reactions as we go. You can think of it like this. This is a graph of like exothermic and endothermic or whatever. I'm essentially doing this. Release some energy. That says free energy. So this graph is free energy. When I drop them and they go from one person to the next, I'm just falling in overall energy. I'm dropping off a little bit as I go. So these five guys right here, these three pumps and these little carrier proteins in between. By the way, the carrier proteins in between are probably insignificant for the AP Bio test. I don't really emphasize them in class at all. I just focus on the three proton pumps. So all of this purple stuff right there is called the electron transport chain. Why? Because it's a chain where we transport electrons. The electrons go from this first pump to the second pump to the third pump. And as they go along, they're dropping off some energy. So picture a candy bar. I'm the first pump. Here are the three pumps. This guy eats a third of the candy bar. He then hands it off to this guy who eats a third of the candy bar. And this guy eats the last third of the candy bar. So we're just, as electrons move from here to here to here, it's kind of like they're falling in energy levels. Again, more complicated than that, but they're overall falling in energy levels to release energy. Every time we move from pump to pump to pump, we're providing that energy to the proton pump. What do proton pumps do when you give them energy? They pump protons. So these guys will pump protons into the inner membrane space. So now I have a concentration gradient. A lot of protons here, not many in the matrix. So where can they go? Well, they're protons. So as we said earlier in this video, protons are positive, protons are polar, and as we said earlier in this video, phospholipid bilayers don't let polar things through very easily. So these protons are trapped. Any protons that are inside of here, they can't really leave. The only way they can leave is through ATP synthase. ATP synthase is the hollow tube. So I've always given the example of your shower. The only way that water ought to be able to leave your shower is through the drain. ATP synthase is the proton drain. These protons have nowhere else to go. They're stuck in here because they can't get through the phospholipid bilayer because they are polar, and polar things don't get through very easily. So they can only make it through the drain. So that's where they go. Anytime they flow through the hollow channel of ATP synthase will spin, and when it does that, it'll join an ADP and a phosphate. The average ATP only lasts for 1 minute. The half-life is 1 minute of ATP in our cells. So within a minute or so, about half of my ATP will break. But that's okay, because ATP synthase can grab them again and shove them back together. As long as there are protons flowing through, this will spin. Well, how do I make sure I never hit equilibrium? Protons get pumped back in by proton pumps. What powers the proton pumps? Electrons. So this is the electron transport chain. Electrons are transferred for energy. The job is to pump protons. So as long as I can steal electrons from food, then I can survive. You could even kind of say the definition of food is something you can steal electrons from. So in class I mentioned this. I guess I can stop saying in class because here's a pen cap. I could eat it. Will it give me any calories? No. Why not? Because I don't have any enzymes that can steal electrons from this. But if I eat, of course, almonds, there are some chemicals in here that I have enzymes for that can steal electrons from this and feed those electrons to NADH. NADH can then send those electrons to power these proton pumps, which pump protons in. They flow out through ATP synthase, and I make ATP. So this will give me calories. As a human, I'm not very good at breaking down cellulose because I don't have enzymes that break down cellulose. So if I eat pure fiber, pure cell walls from plants, like celery is mostly cellulose, then I don't get many calories from it. If I eat a piece of wood, if I just ate a piece of this wood, it's not really wood, but if I ate a piece of this wood, then I'm just going to crap out wood. But if I was a termite, then in my gut, I have eventually, within the termite, there's a thing that is able to break down, that does have an enzyme that's able to break down cellulose. So if I had an enzyme in me that could remove electrons from cellulose, then I could eat, some tree bark, and that would be energy for me. The last thing, again, let me check how I'm doing on time here. All right. The very last thing that I'll mention, and then we'll shut this video down, and we'll come back next video, Krebs and glycolysis. Don't forget to try the free responses that I mentioned at the very beginning of this video. But the last thing that I wanted to mention was, remember in class, I just did it, cyanide. The way that cyanide works is cyanide blocks the electrons from being able to go to the next spot. Ooh, I haven't talked about oxygen yet. All right, so at the very beginning of our next video, I've got to make sure that I mention oxygen and cyanide, and then we'll be done with chemiosmosis, and we'll be ready for Krebs and glycolysis. Thank you very much. See you next video.